Hi everyone, good evening. Uh, can you hear me?
Yes.
Okay. So this is Tanashri from the Dr. Omics Research Lab, and today we are going to continue the next part. Before proceeding further for the analysis, I would like to explain you about the CRISPR. Okay, what exactly the CRISPR, then how we are performing the analysis. That's what I'm going to explain. All right. So, what do you know about the CRISPR? Can you tell me what is a CRISPR? Do you have any idea? Okay. CRISPR-Cas9 system, togetherly we call it as. Actually, it represents the genome editing technology that has revolutionized molecular biology, and it is precise and site-specific gene editing capabilities. It is having precise and the site-specific gene editing capabilities, which are essential, which are essentially allow for an uh, control in manipulating the genetic information of living organism. Means basically this CRISPR-Cas9 it stands for clustered regulatory interspaced short palindromic repeats, and Cas, that's a CRISPR associated protein. It's a gene editing technology which is enabling the precise alteration to the DNA in a living organisms. How does it work actually? Means let's have a look on it. First of all, a little bit history of CRISPR. So actually, it is in 1987. Atsuo Nakata and his team of researchers from the Osaka University in Japan. First, they reported the presence of clustered regulatory interspaced short palindromic repeats, that is CRISPR, in Escherichia coli genome. And this refer to a short repeated sequence of DNA nucleotides, which is found in the genome of prokaryotes, basically. Actually, these sequences are same when we are reading from 5 prime to 3 prime and 3 prime to 5 prime, vice versa. Means whenever we are seeing from forward side as well as from reverse side, these sequences are same only. That's what we call it as a palindromic sequence. Palindromic sequence means what? Like from a both side, it's the same word you are getting. Like from the forward side, if you from starting also, if you will read, and from the reverse side also, if you will read, you will get a same word. That's what we call it as a palindromic sequence. Such kind of palindromic sequences they are getting in our Escherichia coli. Later on in mid 2000s, the functionality and the importance of CRISPR was first realized in prokaryotes. As it turns out, the CRISPR system actually it's a key component of the adaptive immunity, which protects the prokaryotic from attack of viral DNA or a bacteriophage or a plasmid. Adaptive immunity means what? Do you know any example of adaptive immunity? Vaccination, for example, right? So this vaccination, it results in the form of adaptive immunity in a human, since like a body is exposed to the antigens and forms antibodies in a response, which is contributing to development of immunity. So likewise, the same work, the CRISPR system is working in a prokaryotes. So whatever the unique sequences, they are nestled together between the palindromic repeats. Means there are shorts or palindromic repeats we are having, and in between we are having some kind of sequences. Means if I will just show you, how we are having basically this is what like repeat sequences, let us consider we are having. And in between, there are some small sequences we are having in between those palindromic sequence, like this here. This we call it as a spacer sequences. So unique sequences that are nested together in between the palindromic repeats, that we call it as a spacer. These are the bits of DNA that are foreign, but it is not belonging to the bacterium, but instead originate from the mobile genetic elements such as bacteriophages or maybe any transposons or maybe any plasmids that have previously infected the prokaryotes. So these organisms use CRISPR to defend themselves against the virus or bacteriophage, you can say. Actually, this CRISPR system works by the term remembering, means like just like I have given the example of vaccination. Like if we are by the vaccination, we are inserting that particular virus or that particular uh, disease related bacteria or virus like in our cells so that our cells are remembering that, and maybe after attacking, like this is what the example of adaptive immunity. So the same thing or same concept is done by the CRISPR. It works by the system remembering viral DNA sequences from the previous infection and allowing the bacteria to recognize and cut the DNA of invading virus in future encounters. So CRISPR basically it is made up of two key components. One is a repeats and another is a spacer sequence. You can see. So repeats are nothing but what? These are the short palindromic DNA sequence that are identical and separated by a spacer DNA. And spacer means what? These are the DNA fragments from a virus the bacteria have encountered, which act as a memory of viral infection. So these spacer sequence are what? These are the memory sequence, you can say, which bacteria is getting from the viral infection. Now there are another two main components we need to be see about, or we need to uh, like understand about the CRISPR, which is like a Cas9 and another is a guide RNA. So Cas9 is what? So Cas9 is a CRISPR associated protein. What is a Cas9? It's a CRISPR associated enzyme, you can say, or a protein that functions as a pair of molecular scissors, which is working as a molecular scissor, means what? Which is going to cut the DNA. And it can cut the double stranded DNA at a specific location, which is determined by the guide RNA. So what the guide RNA is doing, it is going to guide the Cas9 to bind at a specific location and to cut the DNA or to cut the gene at a specific location. So that's what this Cas9 is acting as a molecular scissor, which is this cutting mechanism is the basis of gene. It's basis for gene editing. So maybe if you want to edit the gene, this Cas9 is going to cut that particular DNA at a specific location where like the guide RNA guided to the Cas9. And next we are having another thing which you need to focus on, that is a guide RNA. So basically, guide RNA, it's a synthetic RNA molecule which is designed to the match, like which is designed to the match the target DNA. Means like it is designed as per the target DNA sequence where we want to cut. So as per that, the guide RNA is going to design. It's a synthetic RNA molecule. It directs the Cas9 protein to a specific location in the genome that needs to be edited. So basically, this CRISPR-Cas9, it's a simple two-component system which is allows researcher to precisely edit any sequence in the genome of organism. And this is achieved by the guide RNA, which can recognize the target sequence, and the CRISPR-associated endonuclease, which is nothing but the Cas that cuts the targeted sequence, and activation of Cas9 protein by the guide RNA binding. So it is going to bind the guide RNA, which is induce the conformational change in the Cas9, and the conformational changes that causing activation of Cas9 nuclease activity. So if I will just show you this image now to just explain how this CRISPR will work. So just focus on this image now. So basically this diagram, it represents the CRISPR-Cas9 gene editing mechanism. Uh, at this core, the CRISPR involves the guide RNA, or you can say a gRNA, which is shown here in a green color. Okay, you can see this is what a guide RNA, which is showing in a green color structure, and it leads the Cas9, which is your cutting enzyme, to a specific pair of DNA. So here you can see the Cas9 enzyme is binding, but where it is binding near to the PAM sequence. PAM sequence is what? Basically, this PAM sequence is one of the unique sequence which is present in the DNA, and near to the PAM sequence only this Cas9 enzyme is going to bind. And once the Cas9 is bind, so which is acting as a molecular seizure, so it is going to be induce this catalytic, like it is going to be it is going to cut the particular DNA at a specific position near to this PAM sequence. PAM sequence, it stands for some photospacer, just a second. Sorry, actually I forgot the full form of PAM sequence. Anyhow, I will tell you later about it. But yeah, near to the PAM sequence, this guide RNA is going to bind. This Cas9 is going to bind, which is ligated to the guide RNA, and then it is going to cut the DNA. So once the DNA is like once we are having the cuts in the DNA, after that the repair mechanism works, because obviously like DNA should be repaired there. So there are two types of repair mechanisms we are having, like a non-homologous end joining and the homologous directed repair. You can see here in the diagram. You can see it's like a non-homologous end joining and the homologous recombination, or the direct repair, you can say homology direct repair, HDR pathway. Yeah, it's photospacer adjustant motif. Yeah, thank you. So what will happen in both the repair mechanism? So in let's talk about the NHEJ, which is like a non-homologous end joining. So basically this process is a error prone and often leads to a point mutation. So here you are getting the point mutations, deletions, or causes frame shift that alter the gene product and eventually its functions as well. So it is favored for genetic knockouts. And this HDR pathway, actually, it's a precise genome engineering method. It's release on pathway here, the piece of DNA that shows the sequence homology to the target site, where it is used to repair the DNA via homologous recombination. And this is a short DNA sequence can harbor or any sort of insertion or alteration, which is allowing integration of any desirable DNA sequences at the target site. So this is what the DNA repair mechanism they are working. And what is our aim here after the DNA repair mechanism? Whatever the DNA we are getting, that we are sending for the sequencing, and whatever the raw data we are getting after the repair mechanism, from that, we are going to find out wherever the changes are occurring in the DNA, wherever the insertions are there, wherever there is a point mutation, wherever there is a deletion. So that finding out the variations, that is our main aim here. So whatever the gene editing data we are having, in that data, we are going to find out the variations. That's our main aim here. How
That's
we are going to find it out? Yesterday, if you remember, I have explained one thing that we are going to align our reads to the reference genome with the help of some tools. And then by aligning the reads, we are just going to match the base at each and every position, and we are finding out wherever there is a changes. We are marking out wherever there is a changes. And that information we are getting in a VCF file format. If you remember, I've explained one thing yesterday, VCF, variant calling format, right? So actually, I have given one data yesterday to download. So it is downloaded in your system. Okay. Uh, Not an issue, sir. I will share the presentation in the drive. So maybe if you want, you can refer that later. All right. So as we have downloaded the data, and I hope everyone is having the Linux in your system. Whoever is attending the session right now, you will have the Linux. So the data is related with the CRISPR. So you can see here the FASTQ files, these two FASTQ files we are having. And right now, we need to be open the Linux terminal from this CRISPR folder. So what you have to do, you have to just select the path. Okay. You have to just select this path like this and type the WSL and enter. So it is going to be open the terminal from the CRISPR folder. So here, if you will do the ls, you are having this zip files, you can see, which we have downloaded yesterday. And now we need to create one script. Okay. We need to create one shell script. So to create the shell script, we are using the touch command. Touch command is used to create any empty file in the folder. And you can give the name as a script, CRISPR script, or whatever name you want to give, .sh extension. sh, that stands for shell script. Okay. So it is going to be open it is going to be just create the shell script. And then here in the shell script, you can write all the commands. But for now, I'm already having the script with me because we don't have that much time where I can type and all everything. So we are providing the script for the reference, and then we are going to we are going to be find out the variations in these two FASTQ files wherever there is a variations, either the point mutation, either the insertion or deletion with the help of reference genome. So, what is the concept here? I will explain you first of all how we are going to find what kind of tools we are going to use. So, let me just explain that so you will get the idea about it. So, let us consider like a first step what we did yesterday, that is a data downloading. So, we downloaded the data from NCBI SRA database, correct? So, as we have downloaded the data, we are having the FASTQ file. So the next step in the pipeline is what? It's a quality control. Like we need to be check the quality, like whether our data is having a good quality or not. So that's what we are performing the quality control. And for that, we are using the fast QC tool. Maybe if our data is not having a good quality, means like maybe if you are having a low PRED quality score, if you are having any adapter sequences or a primer sequences, you can go for the trimming. Trimming, for that, you can use the fast P. Then, as per the discussion in the last session, we need to align our read to the reference genome, which means like we need to be perform the mapping, or you can say the alignment. And for that, we are using the BWA tool. Once the BWA tool is done, after that, in the next step, once the alignment is done, then we are going to be perform the variation calling. And for that, we are using the GATK. So this is how we are going to be proceed one by one. So today we are going to discuss about the quality control and the training. All right. So what you have to do is like as of now you can see you are having this extracted sample, like you are having this zip files and you need to extract it. So for the extraction, because these are the gzip samples, you can see. So to extract that, we can use this command gunzip star.gz. So I'm just going to copy this command, or you can just type and then run, so that the samples are going to be extracted. Sure, I will just provide. Do you have Linux? Okay. So what you can do is I'm sharing these commands which we are going to perform today. Just create a shell script by the touch command, how I have shared, and put all these commands there in the shell script first, and then run one by one. Okay. For whoever is down, like whoever is downloaded sample yesterday, they don't need to perform this wget command. You need to directly run the extraction by using the gunzip. Why I'm not providing all the script? Because you will get confused later. So we will go step by step. I'm sharing each and every command in the chat box. First, add in the script and then run, copy paste it. Okay. So here you can see the samples are extracted. See, this is what the FASTQ files I'm having right now. So once we are having the FASTQ files, after that, we need to check the quality control. We need to see the quality, like whether our data is having a good quality or not. And for that, we are using the FastQC tool. Why FastQC? Because it is providing a very simple way to do some quality control checks on a raw data. It will provide you graph, and by the help of graph only, we can quickly understand whether our data is having a good quality or not. Okay, so that's why we are using the FastQC. And how to install the FastQC? You have to use this command sudo apt-get install FastQC. Once you will enter, it will ask like, do you want to continue or not? So you just have to type Y and enter again. I'm sharing the step 2 also. So just paste it first in the script and then run it one by one. I think I have shared it already. Step 2. So tell me when it's done. Just install the fast QC quickly and tell me when it's done. We can use a touch command to create a script, to create a shell script. Can you see my screen? Here I have created I'm using the touch command and then giving the name script.sh. So it will create one shell script that you can normally open from the notepad as well. Okay. Just right click and say open with notepad, word pad, anything, what text pad, anything, whatever you're having. What is the problem, Anushka ma'am? Tell me. Just unmute yourself and tell me what you're not understanding. I didn't
I'm
move
not able.
any zip files, Sejal ma'am. It was already there yesterday, if you remember.
Hello.
Yes, Anushka ma'am, tell me.
Um, yes, ma'am. So I installed Ubuntu and the root at the rate desktop dash ksct9ml dot what do I write after that?
Your voice is cracking. Is it possible to share your screen, if so that I can check and I can guide you at least?
No, I'm attending this session on Android and using that.
Mm, then how can I help you then? Okay, see, like tell me one thing, you have LTS or WSL?
LTS.
Okay, I got your problem. So if you have a LTS, let me just show you. So whenever you are having the LTS, you have to give some another path. So you have to type like CD, MNT, just type C and user. Okay, so this is what the command you have to type. This is I'm telling only for the LTS user. I'm sharing it in the chat box. Just type it, ma'am, and tell me when it's done.
Okay.
Now, when you will just enter, it is showing you the path. If you will do the ls, so here you can see that it is showing you the different users. So you have to go your username. Like now in my screen, if you will see, my username is just user. So I'm going CD. CD that stands for change directory. So I'm going in the user directory now again. Again, I'm doing ls, and here you can see that you are getting a different options like desktop, then videos, then downloads. So wherever you are having your folder, you can just select that. So let us consider you are having your folder in the desktop. So CD and then desktop and enter. So here you will get your folder. Okay. So this is how you have to proceed if you have a LTS. And whoever is having a WSL, I will repeat it once. Just focus on my screen. You just have to select this path, type the WSL and enter so that it is going to be automatically open the terminal from that particular folder. Okay? Is it clear, Anushka ma'am, now?
Yes, ma'am. Thank
Hmm.
you.
How can I open the Linux? You have WSL, right? So you
Ma'am,
have to
can you
just
please repeat? Ma'am, can you
you
please repeat LTS path again?
LTS path. Okay. If you will just open the LTS, you have to go type CD. If you have a different drive, well, let us consider your data is an F drive or D drive. You have to type like this, okay? CD. Then let us consider my data is in a F drive. So CD F, or if your data is in a C drive, just type that. And then users. Sorry. Just a second.
Ma'am, can I ask you one thing?
Okay. So I'm going with the users right now because I don't have permission to access another drive. So now here I'm doing the ls again to see the list. Now see what is your username. So as of now in my system, my username is a user in capital. So I'm again typing the cd user user. If you may have some another name also because it's depend upon system to system. Again doing the ls, and here you are getting the folders. Okay. If you have a permission for other drives, just select that. All right. If you want to go to the any another drive, you don't have to type users, just MNT and the drive name. Just like here I have given S. Now if I will do the ls, you can see I'm having the folder Dr. Omics Lab. So I'm going with the CD and then again giving the name. So here I'm getting the folders where I have given the data. All right. And whoever is having a WSL, I'm repeating it again, just see it very carefully because now I'm going to start the commands. You just have to select this path, type the WSL and enter. That's it. Okay? I hope it is clear to everyone. So tell me if the FastQC is installed or not in your system. Is there anyone installing? Okay. Anushka ma'am, is it clear now? Like you're saying you're not understanding anything, so now is
Ma'am,
it clear?
I shared photo on WhatsApp.
Um,
As it's
okay,
saying, to disable it, please create the slash root slash dot hash login file. So you are just
you're not having an LTS, you're having a WSL only. Why you're not able to open it from any folder?
I thought I download 20.04
No,
files.
you whatever it is, but you're having a WSL. So you just have to open the terminal like this only. Okay? Is it clear?
Yes, ma'am.
Hmm. What is the problem, Demat sir? If you are having a Microsoft Store problem, that then I cannot help anything about it. You have to fix it by yourself, and then you have to install the Linux. Okay? Hmm. All right. So FastQC is installed.
Excuse me.
Yes.
I'm using Ubuntu,
Uh-huh.
and I have reached the folder where we have the file for FastQ file. What am I
Okay.
supposed to do next?
Huh. Now you have to install the FastQC. I have given this command, right? So just first of all, unzip your samples using the gunzip star.gz. Have you
Mm,
copied the command which I have shared in the chat box and pasted in the shell script?
I have no idea what that means.
See, I have told you now to create a shell script using the touch command. Have you created that now?
Okay, okay, wait.
Hmm.
Mm,
Run this command first.
mm,
Touch script.sh
mm.
and enter.
Okay.
Done.
Uh, yes.
Huh. Now, it's if you will see inside the folder like this, it will
Mm,
create a script file. You just have to open it with Notepad. Done. And once you
uh,
open it Notepad, I'm sharing some commands. You have to paste those commands inside that script first.
okay.
Hmm. I will tell you, Noor ma'am, sorry, how to open the FastQC. Okay, just give me a 1 minute. Now, once you pasted all these commands which I have shared in the shell script, just save it properly by Control S, or you can save it. And then you have to run this command sudo apt-get install fastqc. Fine?
Mm, once I click onto the script file,
Huh?
it opens up in Visual Studio.
That's fine.
Okay,
Just then close this and open it with Notepad. Just right click, open with Notepad, select it specifically. Or even if it is opening with Visual Studio, that's fine. Not an
okay,
issue.
okay.
Yeah.
Okay, fine. Yes,
Now
I'm done.
just paste all those things which I've shared and run the sudo apt-get install fastqc. Okay. So once the fastqc is installed, how to check whether it is installed or not? See, these tools which we are using right now, these are the command line tools. These are not GUI based tools. So you have to check it with the help of command only. So you have to type like fastqc space hyphen help. Okay. If you will see, you have to write this fastqc space hyphen help and enter. So if manual of fastqc is opening, it means it is installed. Okay. Clear?
Ma'am,
Everyone
um,
just focus here. Just 1 second, whoever is talking. Um, like I'm telling this third time, whenever you have downloaded the sample, I'm sharing these commands. You have to first paste this command in your script. You have to first extract the sample. Then you have to install the fastqc with this command. I'm repeating this third time. I'm sharing this again. So please, whatever the shell script you have created, paste these commands there and install the fastqc. Okay? Just if fastqc is installed, just wait for a few minutes. Yes, whoever is talking, just tell me what is the problem. Priya ma'am, you're talking? Like,
I created the file. After
uh-huh.
that, do I have to run it?
Uh,
Uh,
what?
Created the script
Uh-huh.
uh,
Then you have to follow
from
the
the gunzip
instruction.
fast QC.
You using the touch command, we are creating the script, and using the
I have
gunzip,
done
we
that.
are just listen to me first. Using the touch command, we are
Okay.
creating the script. Once you create the script, you have to open that script with the notepad. You have to give this commands. You have to just copy this command which I've shared in the chat box. Like if you will see my screen, you have to copy this and you have to paste it here in the script. Just save it. Now, if you have extracted the samples using the gunzip, then you have to copy this command sudo apt-get install fastqc and you have to paste it here and you have to enter. Then it will ask you for the password. Just give your password, whatever it is, and enter so that the fastqc will install. In your system, as it's a first time installation, it will ask you that you want to continue or not. Just type Y and enter again so that it will continue the process of installation and you just have to wait till it is installed. Done? Is it fine, Priya ma'am? And one more suggestion to all of you, as we are dealing with the Linux and for most of you, Linux is new. So just go through some basic commands at least tomorrow, whenever you will get time. If you will just invest your 1 to 2 hours, at least you will able to get or you will able to understand some basic commands. It will helpful for you. Okay, is it fine? Hmm. All right. So now, as the fastqc is installed, I hope it is installed. Now we have to check whether the like how the quality we are having, like whether it is having a good quality or not. So for that, I'm using the fastqc. Okay, I'm just calling the tool fastqc and I'm giving here the star. Star means what? All. Because I want to check the quality of all files which is having the .fastq extension. So I'm just giving here the star.fastq extension. So it means what? It is going to check the quality for all the files which is having the .fastq extension inside the folder. So I'm just going to copy this command and you can see I'm having a 2 fastq files and I'm running it and enter. So here it is starting the analysis of the forward file. Likewise, it is completing for the first and then it is going to generate it for the second as well. Done. So here it is showing that the analysis is completed. And if you will see inside the folder, for each FASTQ file, it is generated two output files. Out of that, one is a HTML file and another is a zip file. So this underscore one, that is your forward file. For this, it is generated two files. And this ID underscore two, that is your reverse file. So for that, it is generated two output files. So this HTML file, out of it, it is giving us the graphical representation, because this file, it will helps us to understand how the data, like how we are having the data for our sample, whether it is a good quality or a bad quality. Okay, if you are facing a Java error, ma'am, I will give you one command, just run that, okay? Because some FastQC require the JRE. So sudo apt-get install default hyphen JRE. Just copy paste this command as it is, run it and install the FastQC one more time. Okay. And one more thing, just run this command also once. First run this sudo apt-get update, then JRE and then FastQC. Okay. Hmm. Now, if you will see this FastQC report HTML file, this is for the forward sample. You can see underscore one it is. So on the left side, we are having a different parameters on the basis of which we are checking the quality of our raw data. And here, these parameters are showing you the green signs. Some are showing you the red cross mark. I will show you the reverse file also, if you will see. For the reverse file also same thing, some parameters showing you the green sign, some red cross mark, maybe some will show you as a yellow exclamation mark. So what it means that if the parameter is having a green sign, it means for that parameter our data is having a good quality. If the parameter is having a yellow exclamation mark, right now we are not having, but maybe in future. So it means for that parameter, your data is having a warning. It is showing you the warning. It is average quality data. And if you are having a red cross mark, it means it is a poor quality data. But you know one thing, like to decide the quality, that whether the data is having a good quality or not, you don't need to be focused on all of this parameter. There are three main important parameters you need to focus. So what is our first main important parameter? Is this per base sequence quality. Okay. What is the first main important parameter? It's a per base sequence quality. So this per base sequence quality, it will tell you the quality as per the base, like how the Fred score is distributed in overall the sample, that it will give you. Then here the second parameter we are having is the overrepresented sequence, and the last one we are having is the adapter content. So I will explain you all these parameters now, these three parameters one by one. So first parameter, if I will show you, is the basic statistics, which is showing you a very basic information, like what is a file name, what is a file type, it's a conventional base call, then encoding, what machine they used for sequencing, how many total number of reads are there in this FASTQ file. Then if any sequence flagged as a poor quality, you will get the number, but as it is a zero, means we don't have any sequence. Then sequence length, what is the read length we are having, and the percentage GC content. Now, if I'm talking about our main parameter, first main parameter, per base sequence quality. So this per base sequence quality, it will tell you how the Fred score is distributed in overall the sample. And here, this x-axis, we are having a position in the read, that is a base pair, which is like from the first to 289, because we are having a read length approximately 300. And here, y-axis is showing you the Fred quality score. Actually, this y-axis is divided into a three colors, like green, then the middle one we are having as a yellow, and last one, if you will see, it's a red. Actually, in our data, we don't have here, but in this file, you can see the previous, like a green, yellow, and red. So what it is indicating the quality. And this yellow boxes which we are getting over here, like this is box, then this yellow box, these are nothing but your bases. So if your bases are falling under the green area, it means it is having a good quality. If your bases are falling under the yellow region, it means it is having a moderate quality or average quality. And if your bases are falling under the red region, it means it is having a poor quality. Okay. So actually right now, if you will see for our data, uh, all the bases are falling under the red region, and that's why it is showing you the poor quality. I will show you one demo report, which is given by the FastQC page. If I will just go on a FastQC page here, it will show you one demo report to understand it clearly. Here, if I will show you the good Illumina data, how the good quality data will look like. So the per base sequence quality, you can see all the bases are falling in a green region. It means like they are having a Fred quality score above 30. What is the cutoff for the Fred score? 30, right? Greater than 30. And that's why this parameter here it is showing you the green sign. But in our case, if you will see, all the bases are falling in the red region, like nearly 2 Fred quality score. Here also you can see they are falling in a red region. And that's why this parameter is showing you the poor quality sign. It's not a good quality sign. Clear? Uh, what about others? Like, is it clear to everyone? Hmm. Now, if I will show you the second main important parameter, that is overrepresented sequence. Overrepresented sequence means what? Like if any sequence is overly representing in your data, means like you can see this is what a sequence which is overly representing, how many times it is appearing, what is the percentage. So if you are getting such kind of overrepresented sequence, it's up to you that you want to trim it or not, because these are not a good quality sign. But I will suggest you one thing, that if in a possible source you are getting a no hit, if in a possible source you are getting no hit, then you don't need to trim that sequence, because these are the sequences from our data only. But if in a possible source you are getting the keywords like adapter or a primer, so here also maybe if I will show you, if you are having any keyword like adapter or primer, so right now in this case we don't have, but maybe if I will show you this example report again, which is given by the FastQC official page, just to understand it, how we are having, you can see such kind of warning signs also we are having. So in the overrepresented sequence, maybe if you're getting a such kind of keyword like primer or adapter, so this kind of sequences we can't keep. So this sequence you need to be trimmed for sure. But right now, if you will see in our data, we don't have any adapter or primer. All they are no hit. So even if it is showing you the red cross mark, you don't need to be worried about it. No need to do the trimming over here. Okay, it's clear. In our both file, we don't have that. So we don't need to do the trimming. You need to consider both file, forward as well as reverse. Proper structure means like they are not called correctly sometimes. That's what we can say. Means like their base call accuracy is poor. Okay, Unnati ma'am? Yeah. All right. And now our third main parameter, which is like a adapter content. So this is what a universal adapter which can present in your data. If it is present, you are getting such kind of graph. Okay, you can see there is a Illumina universal adapter. They are given with the color Illumina 3' adapter. If you are having a blue line, then if you are having a green line, this is what a third adapter. So right now you can see there is a red color adapter we are having, and adapters are the outside sequences. So obviously we can't keep in our data, right? So this is what the universal adapters, if it is present in your data, in a forward file we are having, and in reverse file, if you will see, yes, in a reverse file also we are having. So this adapter we can't keep in our data. We have to trim it for sure. But from where we will get the sequence of this universal adapter? So I am having the universal adapter sequence of these 5 adapters. I will share it with you now. Just give me a 1 second. So these are the universal adapter sequences. I'm sharing it with you. You can paste it somewhere in some file in your system so that in future also, whenever we are facing a issue, you can use it. If you are getting unable to locate package, so first what you can do, sir, you can update it. So you can write this command sudo apt-get update. I'm sharing the command with you. So please run this command, sir, sudo apt-get update, and then again install the fastqc. Okay. Are you getting my point, Singh, sir? Yeah, okay. And please, everyone, whatever the adapters I'm sharing, whatever the adapters I'm sharing, so please save it somewhere. What to do once the fastqc is installed? You have to run this command fastqc star.fastq, which I have shared in the chat box earlier, or maybe you have pasted in your script as well. Fastqc star.fastq. Okay. Now I'm telling to this everyone, like whatever the tools we required that we are going to install. So fastqc we have installed. After the next tool we require, that is a fastp. So for the fastp you need to write the command sudo apt-get install fastp. This is for the trimming. Then for the alignment we require the bwa tool. Same command we need to use, sudo apt-get install bwa. We require the SAM tools also. So same thing, sudo apt-get install SAM tools. Then we require the Picard tools as well. So same thing, sudo apt-get install Picard-tools. Those, so these 4 tools you need to be installed before our session tomorrow.
Excuse me.
Yes.
Once I have installed the fastqc file, I run the collect fastqc star dot fastq.
You.
It says that skipping star dot which didn't exist or couldn't be read.
Can you share your screen? Yes, Mr. Abid sir, there is a one link.
Okay.
Actually, just yes, you can share your screen, ma'am. And I'm telling to this, Abitsar, as you're asking. Basically, this adapter file I'm getting by the source code installation of the FastQC. I downloaded the source code file, if you will see. Can you see my screen? No, just a second. Okay, I'm sharing the link, sir. You can download the zip file for the FastQC, and if you will extract it in the adapters configuration folder, you will get this adapter list. You can just download the source code file. Is it fine, Abid sir? Just download this FastQC zip file. This is a source code file for the FastQC tool. See, ma'am, just do the
Mm-hmm.
ls once. Just type ls. Hmm. So if you will see, your files are already extracted. So show me
Okay.
the script which you have created and where you pasted the command.
Mm,
You need to show me the script.
okay,
This
okay.
script.sh, just open that file. Show me the
Can you
command.
see it?
No, I think you
Okay,
didn't share your entire screen.
okay, okay, okay.
No problem. Just stop it and share it again.
Mm-hmm.
What about others? Like, are you getting the things? Because um, it's not possible for me to take each and everyone's name. So
Can
whoever
you
is
see?
ready, right. So if you will see here, what is the command? FastQC space star.fastq,
Mm-hmm.
right? So what I have told you that you need to copy this command as it is and then run. But there is some typing mistake you did on the terminal. So copy it, right click copy, control C, control V. Sometimes it will not work.
Okay,
Yeah, right click copy and right click paste. Enter now. I have already given the tools, sir, but still, if you want, I will just give this commands as well. Okay, attendance link is not shared. Just give me a 1 second. I will share the attendance link. Uh, I have shared the tools which you need to install. So please install these tools before the session. Okay?
okay. So, ma'am, is this how it's supposed to look like?
Yes, it's done.
Okay, okay. Thank you.
Yeah. I am sharing the attendance link. Please mark the attendance. Uh, yeah, this is what a attendance link. So that's it about today. Can I download the Ubuntu without? Yeah, you can install the WSL. I have told you yesterday that what commands and all we need to type. So just go through the previous sessions once, yesterday's session, and just follow the process what I have given. Command and all, everything I have given there. Okay. Okay. So yeah, anyone, any doubt? Beginning commandments? Okay, I've shared it. Please go through it. Okay, so Recording you will get from LMS, sir. Sorry, ma'am, Chetna, ma'am. Just see the WhatsApp messages in the group. They have given you the clear guidance, like how to go through the LMS, how to get the recordings and all. So you will get it from there. Okay. Okay. So that's it for today. Tomorrow, just be ready for the install. Like, just keep all the tools installed in your system, and Linux also. Like, whoever is not having a Linux, please install the Linux as well. Keep everything ready so that we can start the analysis directly. And just as per my suggestion, go through the Linux commands, some basic commands at least, whoever is new to the Linux. Okay. Okay. All right then. Bye. Have a great day.